Hello everyone, welcome to the YouTube channel of Chamali Devi Institute of Pharmacy Indore. In this session, we will discuss about histamine and antihistaminic agents.
First of all, we will discuss about histamine. The meaning of histamine is tissue amine, which is present in animal tissues and in certain plants. It is an organic nitrogenous compound involved in local immune responses as well as regulating physiological function in the gut and acting as neurotransmitter for the brain, spinal cord and uterus. It is biological amine which is considered as principal mediator of many pathological processes, regulating several essential events in allergies and autoimmune diseases. The IUPAC name of histamine is 4,2-aminoethyl imidazole. As such, could not found for many years attract enough attention from physiologists, pharmacologists and biochemists alike because of the absence of any therapeutic utility. It occurs in many storage sites in the body in varying amounts. It is present in the mast cells of many body organs, in blood basophils, the mucosal cells of the gastrointestinal tract, especially the acid secreting parietal cells, in the hypothalamus and area postrema in the central nervous system. It is one of the most extensively studied molecules in the medicine, of which synthesis, metabolism, receptors, signal transduction, physiological and pathological effects, there is a large number of collected evidences. The roles of histamine in inflammation, gastric acid secretion, and as a neurotransmitter are the best described role in the human body. Mast cells and basophils release histamine during inflammation. Smooth muscle cells and endothelial cells are the target places of action of histamine. This leads to vasodilation and increase in vascular permeability. In the living organism, histamine is synthesized from the naturally occurring alpha amino acid, that is histidine, by the loss of carboxyl group through bacterial or enzymatic decarboxylation. In this slide, we will discuss about the histamine receptors. Histamine is a bioactive amine which is considered as a key player in the allergic response. Histamine is produced by immune cells but also by bacteria in the gut. Beyond its role in the acute allergic response, it exerts its diverse biological effect through the activation of 4 types of membrane-bound receptors from the aminogenic G protein coupled receptor, that is H1, H2, H3 and H4. This name is given according to their order of discovery. Here we will discuss the role of these histamine receptors. So, H1 receptor. The H1 receptor is all over expressed and is involved in allergy and inflammation. H1 receptor is expressed in many tissues and cells including nerves, respiratory epithelium, endothelial cells, hepatic cells, vascular smooth muscle cells, dendritic cells and lymphocytes. Histamine activates H1 receptor through G-alpha Q11, which then activate phospholipase C and increases intracellular calcium level. As a consequence, histamine elicits the concentration of smooth muscle of respiratory tract, increases vascular permeability and induces the production of prostacyclin and platelet activating factor by activating H1 receptor. Thus, almost all immediate hypersensitivity reaction, including symptoms observed in the skin such as erythema, pruritus, and edema, may be elicited by the activation of H1 receptor. The next one is H2 receptor. H2 receptor is highly expressed in various cells and tissues such as B cells, T cells, dendritic cells, gastric parietal cells, smooth muscle cells, and the brain and cardiac tissues. Activation of the receptor can induce airway mucus production, vascular permeability, and secretion of gastric acid. The H2 receptor is importantly accountable for the relaxation of the airways, uterus, and smooth muscle cells in the blood vessels. Moreover, the H2 receptor is involved in the activation of the immune system such as cytokine production, reduction of basophil degranulation, T cell proliferation, and antibody synthesis. Third one is H3 receptor. The H3 receptor is coupled to G-alpha IO and exclusively expressed in neurons. It is important for homeostatic regulation of energy levels, sleep-wake cycle, cognition, and inflammation. In the upper airways, histamine may play a role in nasal cognition through its activity at H3 receptors. The intracellular pathways related to H3 receptor activation are enhanced calcium influx, MAP kinase, and inhibition of CAMP. H3 receptor can lead to an increase in severity of neuroinflammatory diseases, and it can be involved in blood-brain barrier function. Fourth one is H4 receptor. The histamine H4 receptor is coupled with G-alpha IO proteins and is expressed on a variety of immune cells as well as on other cells such as spleen, intestinal epithelia, lungs, synovial tissue, central nervous system, sensory neurons, and cancer cells. Stimulation of H4 receptor reduces forskolin-induced cyclic AMP formation, which leads to the activation of MAPK and enhanced calcium release. H4 receptor mediates the pro-inflammatory responses of histamine in both autocrine and paracrine manners. Now here we will discuss about antihistaminic agents. The term antihistamine historically has referred to drugs that antagonize the actions of histamine at H1 receptors rather than H2 receptors. Antihistamines are the drugs which compete with histamine at the histamine receptors. These are the antagonists of H1 receptor and serves to decrease or eliminate the effects mediated by histamine released upon allergic conditions like rhinitis, conjunctivitis, urticaria, pruritus, anaphylactic, etc. The therapeutic effect of these agents is mediated by negative modulation of histamine receptors. Classification of antihistaminic agents. The commonly used antihistaminics may be classified
on the basis of their chemical structures. Here, the antihistaminic are classified into two main categories. The first one is H1 receptor antagonist and H2 receptor antagonist. H1 receptor antagonists are also classified into following categories. The first category is amino alkyl ethers. Examples are diphenhydramine hydrochloride, bromodiphenhydramine hydrochloride, diamine hydrinate, doxylamine succinate, and diphenylpyrrolene hydrochloride. Second category is ethylene diamines. Examples are meperamine malate, triphenamine hydrochloride, tonzilamine hydrochloride, and zolamine hydrochloride. Third category is thiophene derivatives. Examples are methapyrrolene hydrochloride, methaphenylene hydrochloride, phenyldiamine hydrochloride, and chlorothene citrate. Fourth category is cyclic basic chain analogues. This category further classified into three main categories. The first one is imidazoline derivatives. Examples are antazolene hydrochloride. Second category is piperazine derivatives. Example cyclazine hydrochloride, chlorcyclazine hydrochloride, and meclizine hydrochloride. Third one is piperidine derivatives. Example phenylidine tartrate. Fifth category is phenothiazine derivatives. Examples are promethazine hydrochloride, trimeprazine tartrate, and methtilazine hydrochloride. Sixth category is second generation non-sedating antihistamines. Examples are tafenadine, loratadine, and acrevestine. The last category is miscellaneous agents. Examples are phenindamine tartrate, triprolidine hydrochloride, chlorpheniramine malate, and cyproheptadine hydrochloride. The second category of antihistaminic drug is H2 receptor antagonist. Example of this category is cimetidine, ranitidine, and oxmetidine hydrochloride. Now here we will discuss the chemistry of antihistaminic agent. So here we will discuss about diphenhydramine hydrochloride. You can see the structure of diphenhydramine, and the IUPAC name of diphenhydramine hydrochloride is 2-diphenylmethoxy NN-dimethyl ethylamine hydrochloride. The molecular formula of diphenhydramine is C17H21NO, and the mass of this antihistamic agent is 255.355 gram per mole. Properties of diphenhydramine. Its hydrochloride salt occurs as white crystalline powder, and its odor is odorless, and it has a bitter taste followed by sensation of numbness. It is freely soluble in water, alcohol, and chloroform, but very slightly soluble in ether. Its melting point is around 166 degrees Celsius. The next one is stability and storage. It darkens solely exposure to light, so it should be stored in well closed and light resistant containers. Uses of diphenhydramine hydrochloride. It is used to treat allergic skin reactions like urticaria, pruritus, etc. It is also used in the treatment of cardiac arrhythmia in combination with entazolene. It is used to treat motion sickness and post-operative vomiting, and it may also be used in the treatment of vasomotor rhinitis. Official preparations. It is available in market as diphenhydramine capsules and diphenhydramine elixirs, and the brand name of diphenhydramine is Benadryl, Caladryl, and Copex D.